Please join me in welcoming to the Distinctive Voices podium, Jason Williams.
All right, thank you, everybody. Isn't it weird to be back in person, but also really wonderful? I'm very, very pleased to have this opportunity, so thank you to Danielle, all the organizers, for giving me this chance to be able to present this with you. So let me start with a question. How many people recognize this and know what this is? I think every hand goes up in here, right? And it's interesting because what I'm going to try to talk about here, at least in the beginning, is, I'm going to build on an analogy, maybe too many by the time I'm done. We all have these documents that identify our culture and our perspective and that are just instantly recognizable. And if you're an American, probably this is one that you would recognize. But whatever your culture, there are documents, there are texts, if I'm using the philosophical approach that Gadamer would have agreed with me on. They could even be works of art that we just find identifying and that we relate to. And tonight I'm here because this is humanity's most universally shared and literally defining text, right? The genome, our DNA, is something that unites every single person. Wouldn't it be wonderful if all of those hands that went up that recognized the US Constitution, that every hand in the world could go up recognizing the genome, DNA, as something that unites us all? So I'm going to be telling a little bit about the story, what's exciting about it, and some different perspectives on it. I work at Cold Spring Harbor Laboratory, which is in New York, North Shore of Long Island, which I'm actually from a few minutes south of there. So one of the few people that work there and actually grew up just around the corner from there. And I also work at a unit within the Cold Spring Harbor Laboratory, which is the DNA Learning Center. And I put there a little snippet from the mission of the center, which is to prepare students and families to thrive in the gene age. And so that's going to be important because that's what I think we're actually trying to do and having some success at. Even though we're just one part of what Cold Spring Harbor Laboratory does, and many of you might know Cold Spring Harbor Laboratory for different reasons in the sciences, we really actually reach a substantial number of students across the world even. Whether it's online, now we're back to in-person as well. But we have this tremendous reach, so it's really a wonderful opportunity for us to be able to extend what the lab has to offer. So what am I gonna try to talk to you tonight about? I've got 3 very interrelated short stories, so that way I'll keep you awake at least for the next 40 minutes or so, maybe have time for discussion and questions. The 3 sort of themes I'm going to talk about are the excitement and the potential of genomics and whatever that's going to mean. And when I get into it, I'm going to show you, I hope, that open learning really can empower that potential and realize the promise that's genomics has to offer. And then I'm also going to try to tell how something called open science enables all of those different things. So I'm going to tell a story, and I'm going to show that it's actually several layers that make something happen, and some of them are not obvious. At least they weren't until I started thinking about it myself. And then, I'm not the only one. Many people, I think, have come to the same conclusion. So when I first talk about genomics, I have not yet been sufficiently punished for putting my slides together at the last moment. I'm always rewarded, and one day it's going to probably fail me, but it's not today. If you've been listening to the news recently, you probably heard the stories about the completion of the human genome, and that is a snippet from Twitter, from social media, from a colleague, Adam Philippi, is a wonderful person, and he and his group have been working tirelessly to finish the human genome. And I could probably answer some questions about that, but I'm going to give a little bit of a history perspective and not so much the details of that story, because that's not mine. But what I would say is that genomics continues to be really important to just about everything that I do, certainly, but it's important to a lot of things in our lives. I just put the 3 of the classic things. So it's important to our food. Obviously, corn is an example. I've had the pleasure of working with people at the lab who've worked on sequencing of the corn genome. It's important to fuel. And this little plant I'll tell you more about, but this is duckweed. So if you've ever seen that green stuff growing on the surface of a pond, it's a really interesting plant, and also some people at Cold Spring Harbor and elsewhere are working to engineer biofuels because it grows so rapidly and it's much more environmentally friendly. If you can get it to produce oil, that would be a biofuel, and then of course pharma. And so obviously the COVID vaccines, which enable us to be here tonight, as we could not for quite some time, that's another example of how genomics really underlies this. So when you talk about the human genome, this is just one visualization. If you go to the NCBI website, I think it's fantastic, and it's one of my favorite things to do if I'm teaching, to open up a web page, and just for all of our students who, of course, grew up before all of this was even done, but it's not that long ago, but they're in their teens, and it's all there. You could theoretically just click on a website, and the whole of this human knowledge is available to you via many different resources, including this one at NCBI. But I'm going to take a little step back to some other points and kind of see how the idea of genomics started, or at least sequencing the human genome. And it does have a tight relationship with Cold Spring Harbor Laboratory. So this is in 1986. And this is Walter Gilbert, who is at the chalkboard, which I recognize. I think we still have the same one if it's the one I think about. Writing out, at these various meetings, all of these meetings where they were planning to sequence the human genome, which at the time seemed impossible. It's very interesting because the human genome took 13 years to the first draft. So 2003 was the first draft of the human genome. You had Bill Clinton shaking Watson's hand, and it was done, but it wasn't really done, right? And so I don't know if he's saying 20 to 30 years is going to be the time that it takes, because if he did, that's really pressing, because it actually just came out a year ago. So then maybe he was completely right. He was on the right track with that. I'm going to go back a little further. Oh, first, I'm supposed to make a joke, if you hadn't noticed. I love the choice of microphone that he has there, and the technology, which has obviously changed, so we're not dragging a whole microphone. That would be uncomfortable. But in no pun intended, miniaturization is going to have a small part in my talk a little bit later, the technology has moved on from there. But I'm just going to go back just a little bit further, and I'm going to go back to Theodore Boveri and his wife Marcella. And if you don't know them, they were working as embryologists. And Boveri was pretty much one of the first people to come up with really a theory for cancer, because he realized when they were working, in their case, with mostly sea urchin embryos, they started to realize that the segregation and the movement of chromosomes could lead to these types of defect in cell division. And he started to formulate a theory that, in fact, if the number of chromosomes was not correct, that would lead to deficits. And in fact, maybe even cell division required something that the chromosomes had, and that maybe a signal needed to be overcome before a cell would decide to divide. And so he came up with this theory that the wrong combination of chromosomes could cause problems, and eventually is really a foundational idea that later really came to be our understanding of cancer. So he might be one of the first ones. He certainly was part of the generation that rediscovered Mendel's laws, but he really is a nice beginning, convenient for me tonight to say, is a person who thought that DNA, that chromosomes, they didn't have DNA at the time or exactly know what it was in terms of genetic material, but they thought it was important. And then obviously we know the story goes on from there, right? You had Watson and Crick and Franklin. That's another point in the timeline. And then there's also, this is one of the early Cold Spring Harbor meetings, and you can see people like Francis Collin, and Watson is in that photo. So, I'm just going to draw that chain. But let me also draw your attention to an undercurrent of something's happening here. Maybe not a way that the story has been told before, but the way that I'm going to try to tell it tonight. Back with Boveri, you have, a small number of people, maybe him and his wife. I don't really know if they had lab assistants. I'm unfortunately not a historian of science, so this is my understanding. But you had, a relatively small collaboration that depended upon a microscope, and a lot of good biology has been done that way, 100% for sure. Next, when we're going up in levels of sophistication, when you have, the double helix, you have Watson and Crick and Franklin. Now you're talking about bigger teams and other sorts of things that needed to be in place and that you might not think about that needed to be in place. X-ray crystallography had to be in place. Scholarships that would allow an American to go study at Cambridge, that had to be in place. There are all sorts of things that needed to be in place underneath that were potentially, not sufficient, but at least necessary to those steps happening. And when you get to this Human Genome Consortium, the diagram that I have there is actually a diagram of the software that was needed in order to do the data analysis. And this was maybe one of the first times where DNA data needed to be computable, and you need to make use of computational power and software tools in order to actually accomplish what you were looking to do. So I'm going to say that these ideas of computation and collaboration and openness are really essential undercurrents to what is needed. And it might be, if I'm right, that if you don't pay attention to those things, or you don't really take full appreciation of those things, you might miss out on some opportunities. And you can guess, I am saying we're not probably taking full advantage of these things and not doing everything that we could. And there's some fantastic opportunities there. So as a person who really is, as that maybe the introduction suggests, and pretty computational, I'm going to say the future is computational. And what do I mean by that? Well, I think it's a great time to be a biologist. There are so many different tools. Now, I probably referred to this a couple times. The DNA sequencing that took the whole globe of scientists, all the people that were involved in that consortium, can be literally done in the palm of your hand. But it's more than just DNA data. We have all sorts of insights from imaging and neuroscience. We have now this really big computational layer, which I'll talk a little bit about. And all of those things rapidly lead to something like the COVID vaccine, which I'm going to hit on many, many times just because it's in everybody's head. And it's just a super example because I think it's only the beginning of what we had. A few days ago here at the Convli Symposium, we had one of the persons from Moderna who was responsible for doing this. And as many scientists are really appreciating, who've been doing this for a number of years and working towards this moment, COVID happened to be the first one through the gate, but there are many, many more opportunities that are going to be enabled by that. The other thing is that, biology is a data science, and we hear that term all the time, but, nobody, for those of you in the audience who might be biologists, there's no pipette biologist or microscope biologist or cell culture biologist. You haven't heard of these job titles. We're all just biologists. And so that's because it's the questions that we can address no matter what the tools that we use, even though computation is one of those really important tools, and I argue is becoming more important. So we've got a problem, though, with that. And the problem is, for most biologists, I will say, and I'm cautious about saying that's a problem, but then when I say it, all the biologists shake their head and say, yes, it's a problem. The problem is that most of us as biologists went to school for something like this. And then when it comes to doing our research, increasingly, we need to know some of this stuff that's on the right-hand side. And so there's a little bit of a divide. And it's an interesting conundrum that I find it interesting. And it's something that is, again, a place for opportunity. The transformation that all of this happening is I'm going to say, recent and rapid. So I'm going to pick on the astronomers. 2 times is the first time, but this is good stuff. The first telescope, what you could argue is the first telescope, was actually Lippershey in 1608. And Galileo's was based on what Lippershey designed, so I'm trying to give a little bit of credit. And I just picked a convenient time. If you move 400 years into the future, you have things like the Kepler telescope, right? So you have that gigantic change in what astronomy meant and the instrumentation that enables that astronomy. And even, data was taken probably on observations on paper versus on a hard drive. That was a 400-year process to get from A to B. Now we have James Webb, we have all of these other things. So that was a really That was a decent amount of time. In biology, the equivalent of that has happened in like around 40 years. So the very first nucleotide sequences, which are actually RNA, came before DNA, maybe in life too, but and also the data. We've got 1960s where we start to get our first sequencing. Sanger really was still the same technology that we used for the, at least parts of the original human genome. But to go from that to some of the first, what we call high throughput or next generation sequencing machines, was a 40-year transition. So imagine if you were an astronomer and you started your career with the equivalent of a little brass telescope, and by the end of your career, you needed to be able to use the Kepler. That's an amazing transition, but you have biologists who are still practicing who went from this to this. So that's a really amazing transition. So that just suggests to us that really the skill set of what's happening in biology has undergone a drastic change. And I think we don't want to belittle that, because it means that we need to do some extra steps perhaps to take advantage of what's possible. Some of you may also have heard of one of the most important breakthroughs. It probably also could be worth a Nobel Prize in the near future, and that's something called AlphaFold. I'm not going to get too deeply into it, but one of the most important challenges in biology today has been protein folding. DNA and RNA are kind of simple. Most of the basics are worked out in the 60s and 70s of what the genetic code was. But if you take a piece of DNA and try to figure out not the sequence of the protein, but the actual real shape of the protein, it's a problem that you can throw supercomputers working at it for years and still not know that you're correct. There have been other tools, and there still are, like cryo-electron microscopy, but all of a sudden you have this AlphaFold where you're using machine learning and AI to generate really accurate predictions. This is going to be tremendously important. It doesn't solve every single problem, but that is going to be tremendously important because that's how you design drugs. Most drugs are targeted to a protein. You need to know exactly down to the femtosecond how little molecules are moving on that protein in order to effectively design a drug. But if you look at the number of papers in the field using not AlphaFold, but machine learning and artificial intelligence we've been hearing a lot about, it is basically a hockey stick inflection, where, starting a little bit after the, so let's say around 2010 or so, it's just gone up from nothing, which is, of course, what you'd expect before, to 10,000 papers that are referenced, in this case on PubMed, that use these technologies. And so when every time I discuss this with other biologists, they're like, yes, this is nerve-wracking. Because what do you do if you're a biologist and you started with your Lippershey or your Galileo telescope, and now someone's saying, okay, you got that, now you need to be able to operate the James Webb Telescope. Would you be able to do that? You'd probably need some significant training. But we don't necessarily provide everything you need, I think, in my opinion, so we're going to go into what. But that skills timeline, again, this is maybe a fan service for any biologist in the audience. When you're a molecular biologist, again, that's what I grew up with. We had all of these technologies for replicating DNA. We had wonderful things like being able to examine how much RNA is produced in a cell. Now we have things like CRISPR, which is going to be fantastic for gene editing. But as a molecular biologist, these are all in the same line of skill set or reasoning. So I have not done anything with CRISPR. I do a little bit of work in the lab, but not too much. But if you threw me in the lab, 2 weeks later, I will have CRISPR working. Just give me the reagents and give me the kit, and I'll read a paper or two, and I'll get it working. But if you think about the other skill sets, the computational skill sets, you started with word processing, which at that point probably just meant securing time on the typewriter to type out your thesis, to going to saying, oh yeah, you need to be able to deploy a Hadoop cluster on Amazon Lightsail. It's a gigantic skills gap. There is really not a nice path that can take you from really basic computational skills, or what we consider sort of okay, I can learn how to do this, to things which are really, really, orthogonal to the training that you grew up with. So there's still one more problem yet. If you believe it, before I try to get to solutions, I promise you there's lots of things to be optimistic about it. But also the data challenge are huge. I always like this paper, which suggested that we shouldn't use the term astronomical to mean big. We should use the term genomical to mean big. This graph here, which quickly has gone up into numbers, and I'm not confident I've got the pronunciation right on or the math. It's something like 63 quadrillion bases of DNA sequence that's housed at the NCBI sequence read archive. So the amount of data that biologists have to interrogate is absolutely, beyond the ability of an ordinary person to comprehend. To tighten the screw just a little bit and make it even worse, well, this time I guess it is good for the astronomers and bad for the biologists. In astronomy and physics, I think that one advantage they have, because they also work with big data, is that at least in order to do big things, technologically, their platforms and their tools have converged. So there's not 20,000 different Large Hadron Colliders. There's one Large Hadron Collider. Yes, it has different experiments running, but lots of people are at least using the same machine, so there's an opportunity to converge on, what needs to be done, what procedures, what methods, what protocols, what training. There is a convergence there. And the same astronomers, right? They have put all of their things into a way that at least converges some way. Biology, of course, naturally is going to do the opposite. It used to be when we would go to conferences a couple years back, but not too long back, some of those earlier big sequence machines, by big I mean the size of like, a washing machine, it would be that you go to the conference and you'd be curious to look at the latest update of the map of who owned one, and it was country by country. Now again, this Oxford nanopore sequencer is literally smaller than my cell phone. Can fit in my hand and can produce just as much data as those machines could. So it's become, I'm going to argue, it's become more decentralized because and it's very good. The technology is now open. Everyone can have that technology, presumably, and produce in their own lab what used to take a global consortium to do. But it does mean that everybody can kind of go off and do their own thing. So you can do it differently, someone else can do it differently, and there may not be as much of an opportunity to converge on consistent methods and training. So there are things that can happen. And to me, what that started to evoke, all of these problems together, it started to evoke the idea for me of this sort of a scientific digital divide. For those of you familiar with the term, I don't want to misappropriate it because the term digital divide is really talking about bigger socioeconomic issues than just science, but it's really the idea that some people would be connected to the internet, some people are not. Some people have computation, have a computer, and have access to those things, and some don't. We really saw this during the pandemic. I didn't have time. I'm sure, way too many slides here. Not to scare you, most of them are sort of the animation, so it won't take that long. But one that I had to leave out was just the idea that, I think it was in Massachusetts, the number of underrepresented students who used to score at the top achievement level for one of their standardized tests. There was something like 900 of those students a year you would see who were underrepresented, Black, Hispanic. And then after the pandemic, it was something like a fraction, 10% of that. Because all of a sudden, hey, if you came from a neighborhood or from a household, you didn't have that computer, you didn't have Zoom, you didn't have a webcam, right? The digital divide. But I think there is a divide in science, or the potential for divide, when some scientists have these training and have the skills that enable them to use these tools and some don't. And it really is real, I'm convinced, because when I talk to especially early career faculty, people are starting, what does it mean if you're going out as a scientist for a job, you're trained, you're an expert, fantastic biologist, but you don't know any machine learning, and the other person who did get that training somehow does it, and you're both going out for the same position. Can you do the same science, right? It's an interesting question to me, and it's one I think we should pay a lot of attention to, because you're in a position where you could potentially have schism, a divide. I don't know what to call it yet, but you can have schism, and I'm referencing schism and putting it against heresy, mostly because I wanted to use this quote because I like it, and I don't like schisms. And it's a quote from a theologian who said, if you must make a choice between heresy and schism, always choose heresy. And what he meant by that, and it's kind of a pun because heresy actually means choice, is that we shouldn't be forcing people into vision. We should be giving them a choice and really working to serve the needs that they have. Now, one question that I think, in at least arguing with myself, and maybe listening to you later when you have some thoughts to give back to me, is this really a new problem? Doesn't every generation of scientists need to keep up? Every generation of scientists, they're going to have something new, and is a bit of kind of complaining. Well, to reference who I quoted twice removed in my abstract, Planck said science progresses by funerals, and Kuhn agreed with him that, hey, the people who really have an idea and they won't let go of it, it's not necessarily the right idea, but, the department chair and you really can't say anything. Once they leave, the new ideas will come. It also reminded me of, you can imagine that the quote-unquote curmudgeon who doesn't want to give up their ideas, but is worried about what's happening. It reminded me of a quote that I had to dig up from one of my favorite composers complaining about his students. And Arnold Schoenberg said that his students were always trying to outdo what I offer, which puts me in danger of becoming their imitator. So yeah, there's always times when you could look at the problem and you could say, well, that's just history and that's just natural. The next generation will come and things will move on. But I argue that we shouldn't be so dismissive of the idea that we have a divide forming. Because, and I'm going to rely on Twitter a lot increasingly, I really like this article in Wired, which talked about machine learning, that it's not just doing something better, it means doing something humans can't do at all. And what starts to happen is that we're moving into an area, science has its stratifications and it has its layers of people who make decisions and who, make the field work. But if you reach an area in which the people who are making the decisions and the people trying to proceed and decide how should we make best use of funds, how should we decide how resources are allocated, and they don't have the type of comfortable grasp on the science as they would like to, or if you are an investigator and you actually can't understand because there's a knowledge and skills gap and even a language and terminology gap, between the science that's happening in your lab or in your discipline and the science that is happening that your postdoc or your grad student is doing or your collaborator is doing and that you can't, that becomes an issue. It's something that we should be working to solve. This was a report from one of the scientific, there's a whole research and investigation paradigm around people who are concerned with scientific software and increasing its reliability. And this is just a quote that I found interesting, saying that there's software that all of us scientists use. And what happens is that this software gets used by everybody. So it has a great impact on the field. And there's another paper I could pull, which if you look at the #1 cited papers of all time, the top percentage of cited papers are always method papers, because people cite methods when they're new, at least, because it's important. But the systems that we have to sort of review something like software, that's actually something that hasn't kept up. So in other words, if you are refereed a journal publication, you're reviewing something, obviously we focus on the science, but most of the scientists who will be reviewing a paper that relies heavily on a software or relies on one of these technologies like AI or ML, they may not know very much about it. And so they're kind of forced to take it on faith that everything is happening the way that it should. Yes, some things you can sort of suss out without having full knowledge, but are we underserving ourselves and also our reviewers, our funding agencies, by not making sure that we have all of them supported with the training and infrastructure they need? Because what happens is that you're changing incentive structures and you're balancing things in a way that, or unbalancing things in a way that they haven't been before. So what should we do? Am I done with problems? Well, just a few more. One, this sort of leads me to the idea that we've got to work on scaling ourselves and improving what we have to offer people, because the same brain that we had when this all started, let's say in the 1980s, started to get dangerous with some of these technologies, to 2002, we have exactly the same brain. And this is not just my thinking. When we asked scientists, so this is work that we did a couple of years ago, asking scientists, this case, investigators who are funded by National Science Foundation and Biological Sciences Directorate, we asked them, well, what did they need in order to come into this world of big data or do research? You don't need to read all of the little font and fine print. The key thing that all of them agreed is that they need more training. on how to work with these data sets, how to integrate multiple data sets, how to work with data management, how to scale their analysis to the cloud. So the scientists are really already sensing that this is an area of critical need. Going back a little bit deeper to how those scientists got trained in the first place, or how we're doing today, one project that I work in, one of the things that we found there is that most undergraduate faculty believe that this is an issue that we should be teaching our students, we should be doing a better job of it, but only a fraction of faculty actually report that they bring these types of experiences and training to their undergraduate students. That's already a big disconnect, which means that the chance that we're graduating undergraduates now who are prepared for this, we're still not there yet. And in fact, when we actually look at who is doing the teaching and the training, it turns out, maybe counterintuitively, the faculty that were more recently graduated, those who got their degrees a little bit later on, they're the most likely to have formal training. Almost half of them have at least some formal training, maybe not necessarily a degree in bioinformatics, but they have some formal training. But when we ask, well, who of them are actually bringing it to the classroom, those who have the most training are actually least likely to bring it into the classroom. And we think that's because, to quote an article that I liked, I don't think it was the New York Times, it was one of those other type of headline publications, students love science students love math and science until they figure out it's hard. If you are a new faculty member and you want to introduce a new course and you want to introduce new hard things to your undergraduate introductory courses, you are also concerned about, tenure reviews and student evaluations of your course. And am I doing this right? And do I have enough tools? So there may be some perverse incentives why we're not bringing this into the classroom at the rate that we think. Other colleagues at the DNA Learning Center, namely the executive director and founder, David Miklos, and one of our evaluators, Lindsay Barone, they showed that it used to be that NSF, National Science Foundation, poured a lot of money, especially during the in the early 90s. There were a lot of teacher training grants where NSF really supported teachers and giving them new experiences and training. This is actually on the high school level, so I'm going back further still, so that they could be equipped with the latest idea of what science is, the latest tools to bring into the classroom to inspire their students, and that funding has since sort of trailed off. So we are at a place where there's a lot of, I'm going to say, be very positive, there's a lot of opportunity for us to do better. So how might these issues be addressed? Okay, well, I think that we need to talk about empowering these people with open learning. And what do I mean by that? I'll elaborate and give you some examples from the DNA Learning Center, what we're trying to do and others in order to make this better. One of the fundamental principles that the DNA Learning Center has operated on when it comes to doing these things is the belief that, especially in biology, it's true, that students can work with the same data at the same time and with the same tools as research scientists. And usually the students, you throw a really hard problem at them, they're going to work for it. It's the teachers sometimes are a little bit nervous about doing it. But if you don't tell the student that this is a problem for an undergraduate and they're a high school student, they're going to work at it too. And they'll be bright and clever and get through it. I like to have videos and animations at some point in my little presentation, and who doesn't like liquid nitrogen boiling away? This is those little duckweed plants that I showed you. When we grind them up, then we can prepare the DNA. We did this last summer with our high school students and start sequencing the DNA and assembling genomes. These are the same tools that I'm using with high school students that are being used by every consortium, that the whole complete human genome, that depended upon this nanopore technology. They had many more of them, but we had one, and that was enough for me to get high school students using the same technology as anybody at NIH or elsewhere in the world. One of the other cool things that we've done to really bring hands-on training is this project that we've worked in a number of years in Long Island and also especially New York City, where you can imagine there's less resources, fewer resources to work with students, is this idea of DNA barcoding. And very briefly, DNA barcoding is the idea that you can take an unknown food, an unknown plant, an unknown insect, and extract a little bit of DNA in a very simple experiment that could take an hour or so, get that DNA, sequence it, and that takes a day, and you can actually generate some interesting results. And the students put together their questions. When we did this is back in 2012 when we started, but we're still doing it. These students were the first winners of the little competition that we had. And what they were looking at was ginkgo products. So they went to the CVS, they went to the herb medicinal shop, they went everywhere that was selling ginkgo supplements and extracting DNA and looking to see what they could find. And as you can guess, they found no ginkgo in any of the samples that they were looking at. They could only find rice powder or like grass clippings. But if you think about it, if you buy a capsule or a tablet or tea or seed or something like that, you might, there's no way to trace that back by visual cue that that's actually ginkgo or not. And when they used one of the tools that we built and looked at it, they were expecting, this is the ginkgo sequence up here with all of these marks, and all of their samples is not coming up with any kind of match to ginkgo. And they weren't the only ones that found it after that, after they did their experiment. The New York State Attorney General also happened to find that that was true, and so they had to have cease and desist notifications to Walmart and others that, Now, and they're not saying that any given company or group or person or store actually even knew, because you might not have known that what you were selling was not in fact what it was. But it's just one clue that, one example, if you give this opportunity to students, they can do the same type of research as anybody else. And the cool thing about these types of things is that these experiments are fairly simple. They're really hands-on engaging. They combine the wet lab part, which students love, with the computer and computational tools, and they scale. So we've worked with thousands of students in these various projects that have been funded by various foundations, including NIH. We had the Sloan Foundation money and various other sponsors to be able to generate thousands of sequences. And in fact, many of these new sequences that had never been sequenced before get put back into the public database so that other scientists can use them. One of the programs that I work with is also to get underrepresented students into science. And so we put together this biotechnology and bioinformatics camp and alumni work where we work with the students over the summer. And then we also mentor them and put them into a network so that they can help each other. They have, going back to that mission of educating students and families, their families are required to come to the orientation so they know what their children and family members are going to be doing. We go and do labs, they have speakers, we go to college for a day, and then they present back to their family and friends. They have a requirement to give back to the community. And so we had projects where, in this case, we were trying to talk about how to combat vaccine hesitancy and misinformation. And so they did interviews with different institute directors from NIH. They made their own videos, animations. All of these things, students can do amazing things if you give them the tools. The tools and the infrastructure, which I work a lot about, was matched on, are NSF-funded research infrastructure. So all of those things, like those tools to look at the ginkgo samples and be able to do the DNA comparisons, that's powered by high-level infrastructure that is the same infrastructure that scientists are using to assemble genomes. This infrastructure is used for a whole bunch of things, even the black hole photos, or all of these sorts of different things. We can give students the access to that and make it possible. The other thing that we try to do to try to combat the issue that we have there is really make recommendations. And one group that I'm also involved with, we try to provide the types of recommendations that teachers could think about by providing them with competencies that should be working for in their curriculum. And with another group of collaborators, we really talk about, in this preprint, the idea of doing a National Center for Science Engagement, because with a modest amount of funding, we could have students at the undergraduate level really doing experiments and hands-on science as a matter of course. I can't go into it because, of course, they don't have enough time. We know that if you have students, especially in their freshman year, doing hands-on experiments as part of their regular courses, not the extra summer course, not the internship, because those don't scale. We'll reach a few students, but we won't reach them all. We improve through hands-on engagement we improve graduation rates, we improve retention in STEM, and we're authentically training those students for what science really is. If you go even beyond the undergraduate, I'm working my way back up to, scientists who have been trained for a little while. At a different level, one of the strategies that people have been employing to get the graduate students and get early career faculty up to speed is by throwing them into a workshop and saying, okay, you know nothing about machine learning. Come with me for 2 days. I will give you a machine learning degree. It turns out that doesn't work very well in most cases. This paper that was in PNAS appropriately for this location, they showed that when you actually look at many of these short interactions with these skill building, it's not as effective as you might think. And we spend literally tens of millions of dollars between at least just NSF and NIH funds trying to do this, but it's not as effective. There are a lot of reasons why. I probably won't go into them right now. I'll tell you what we're doing. But this idea of career-spanning learning is really important to me, at least, and to many others, I think, because I think that we have wonderful, bright scientists at every level of career, and we should be supporting them so that no matter what their stage of career, wherever they are, they should be able to get access to skills they need and repurpose and reinvent themselves to face the challenges we have today. We saw that during COVID, right? But during your formal preparation, you're guided by a curriculum, you're supported by lots of learners who are learning the same thing, and everything is sort of mapped out. Once you leave your PhD and you're out in the world, yes, we're all scientists, we're all lifelong learners, but you're on your own. When you encounter new things, they're untranslated, and probably the most important thing is your own time. Because if you make mistakes learning the wrong thing because somebody told you should learn Python and learn how to code in Python, and it turned out you should have learned R, well, now you've wasted a long time. How do we make that transition easier? One funded project I'm working on is a project to make career-spanning learning effective and inclusive for everyone in the life sciences. And we've come up with and working on a framework of core principles to improve professional development for life scientists, to try to make it more inclusive, effective, catalytic, and based upon really good pedagogy, and then be able to scale good training that meets those criteria and make it shareable, sustainable, and scalable. So we're working on that. Another thing, a project that I had worked on and continue to do, is to build community of practice. So there are people all over the world who are working on developing skills for life scientists, and we have an online community so that we can learn from each other. So the last part of my talk, and reaching up the conclusion here, is open science, because that's the other thread underneath this open learning, this idea that there are practices and principles and even technologies that make all of this possible. So what is open science? If you're, as a term you've only heard of, you're not quite sure, because it is a bit fuzzy, I think, sometimes. This is a nice graphic developed by some colleagues, and they mention a whole bunch of things. Open data, which is the ability to really make your data accessible to the scientific community. It's kind of shown right now, I'm not going to reference it, that when a paper says something about data available upon request, it often isn't. And so making sure that the data is available. Open educational resources are kind of like some of the things I mentioned, trying to make sure there's no paywalls. I ran into that in the hotel room. Peer review and methodologies, and open source, which is the software code that often goes in and behind science. And interestingly, these principles and practices, there's not too many open science courses. You won't go to a graduate school very often and find a course called open science. This is all things that are either learned on your own, or increasingly people are developing training because it hasn't been trained before in a formal sense. I'm going to quote, again, a friend from Twitter who basically will just prove my point by his tweet. I didn't ask him to write exactly that, but if he wrote that spontaneously, I think others feel the same, that just about all of our progress relies on some of these open science principles in action. And it's what allows us to progress. So if you knew about bioinformatics, or you've heard about that and heard about data science, but you never really heard about open science, it's a gap in awareness that we need to work harder to fill to make sure people know about the benefits and the importance of it. Just to give one or two concrete examples, and I got to move on as we wrap things up here, that AlphaFold, which I mentioned is a really great breakthrough in machine learning and AI, that's only possible because all of that data is open. And biology had a lot of early pioneering open science practices that were embedded from the beginning. And so we don't want to be sort of blown away what machine learning and AI can do without also acknowledging the work of many, many people who worked already to make that data open so that it could be done. Another one, which sometimes brings shivers to people who existed in one paradigm of scholarly communication, are the idea of preprints, which is the idea of before you even submit it for peer review, it being open. And isn't that dangerous? And should we be afraid of that? Preprints are nothing new. This is going back to Francis Crick and one of the first preprints, where preprints came from biology, not physics, although the physicists did a much better job for a while in curating it. And it's something that's been really important, and COVID is another example where 32% at this point of COVID papers were preprints first, and the average would have been 3. And if you think about it, I remember taking this slide at the beginning of the pandemic, that when in history did you have a virus where all of a sudden people identified the virus, sequenced the virus, looked at the phylogeny of that virus, created tests, and did all of these different things within a week later, right? And all of this was enabled by even social media as part of it, where people were able to share those data immediately as they were coming out and work on it. That would not be possible if, under the traditional way of doing it, a journal may take anywhere from 200 days to get something out, versus something on bioRxiv is out on average in 2 days. So open science is not new, but it evolves. It's something that we have all of these different new tools that support it. There's more tools yet that support it. When you go outside of the sciences, these are all just logos. You don't need to know what they mean at this moment, but there's the idea that there's a whole ecosystem of technologies that really have enabled this, and most biologists might not be aware of them, but you should be one day. The other thing about it to acknowledge is that it's incredibly fragile. Because a lot of the tools that we actually depend upon, these are 3 really, really important scientific softwares that have literally millions of downloads and rely mostly on volunteer effort, 5 people who maintain this. This is an earlier slide. This is from 2017, but although I'm sure the numbers have changed slightly, this is a pretty common thing that you see. So if we want to move from what we were able to do in the time of the Human Genome Project to now, when we have projects that want to sequence everything, the data is going to get more complicated. The teams, thankfully, are getting more diverse. We're definitely going to have to think about that underlying current of open science. We're definitely going to have to work to reduce the barriers to open science, really helping people to understand different perceptions, answering questions, because scientists are concerned about getting credit for their work. There's been work by national academies and others in proceedings trying to solve some of these problems. But you can just enable cool things. I threw it out there on Twitter, hey, I'm giving a talk on open science, talk to me and tell me something interesting. And one person poked up and said, hey, I'm a doc worker with a New York City high school education, and I submitted 2 reference genomes to NCBI, 3rd one coming soon, and it's there. That's amazing if people can participate in sciences because they have those tools out there. It can be scary, but it's also amazing as well. So I'm going to wrap up by really saying that open learning and science is a critical priority. It's something that you might not have thought of being underneath all of that scientific progress, because it's not the Nature publication, it's not the 1 or 2 scientists who get all the acclaim, it's this whole ecosystem underneath. And I want to finish it also, I'm saying finish it, I've got 2 slides, with a tremendous opportunity. This came out Friday. This is from the Pew Charitable Trust, so you know them as doing survey researchers, and it was a survey of, in this case, Black Americans, Black adults, and how they see science jobs. And science and engineering, the perception is that those are the least welcoming and least opening to Blacks. That's what this Pew study. So what an opportunity to change that perception. So with that, I want to thank everyone for their time and attention, and really hope that it gives you something to think about. And with that said, I'll take a couple of questions. So thank you so much.